Philip asked me to talk about microbial metabolites and how they can interact with the broiler gut wall to improve performance. Well, first of all, I should say what we know about microbial metabolites, that's what I'm going to talk about, but there's a huge amount that we still don't know, so keep that in mind, please. There was a paper out actually in 2015 that predicted that antimicrobial use in food animals would increase by 2030. Fortunately, I think this prediction will hopefully and probably be wrong, because we see that very recently there's a move all over the world to stop using growth-promoting antibiotics in animal feed. It used to be only Europe. Some crazy politicians in Europe decided in 2006 to stop this use of growth promoters, but now this seems to be worldwide. And this is probably one of the major reasons why we are sitting together here today. Indeed, we have been, in the poultry industry, we have been probably spoiled for many years using growth promoters to sort of alleviate any possible problems due to shifts in the microbiota composition in the intestinal tract, following the kind of nutrition or the changes in nutrition which are being implemented. Microbes were not of interest to nutritionists at all in the past, to the point even that they used ileal fistulation to study the effects of nutrients, completely ignoring what was happening downstream below the ileum. So cecum and colon were of no interest at all. And now it seems to be the other way around. Cecum and colon seems to be the focus of everything. But anyway, when we come to think of it, Dr. Moore already said earlier on that chickens are coprophagic, so it seems quite logical that they pick up, on average, around about 1 million bugs per gram of anything they pick from the environment, but also, this is on average, taking the average of drinking water, feed, and picking the environment. So it's probably a little bit lower in humans, the number of bugs we swallow per gram of whatever we swallow, but still we should realize that any animal, including humans, are ingesting quite a number of microbes all the time. And the thing is that, in principle, of course, bacteria are cells built of building blocks that are essentially similar to the building blocks that are used to build eukaryotic cells, which is amino acids, nucleic acids, and you name it. And the consequence of that is, of course, that to some extent we may compete for the same nutrients. And so in the intestinal tract, this is a problem, because you don't want your competitors to run away with your nutrients. So, in the animal, in the intestinal tract of the animal, it is clear that the crop and the proventriculus play an important role, because we know that there the feed is acidified and the pH drops to about, say, between 5 and 6 in the crop, and a lot lower again in the proventriculus. And it was always thought that this was meant to kill the microbes in order to have a more or less sterile environment. But I personally don't believe that anymore, because if you want to kill microbes, you don't use acids. No, to the contrary, you preferably use alkaline, because that's a much better disinfectant. But what acids do, actually, they do kill a number of microbes, but other microbes, actually, by getting into contact with acid, they enter into what is called a viable non-culturable state. And the viable non-culturable state is actually a situation in which the bacteria still are alive, but they cannot multiply. They can survive, but they don't multiply. And that's the trick nature plays to get the small intestine, especially at the level of the duodenum and jejunum, not sterile, but at least in a situation where there's no competition for the nutrients. The number of microbes that are generally present in duodenum and jejunum are around about log 3, which is the concentration of microbes you get on average in tap water. So it's quite clean. But of course, the microbes actually can be controlled like this, kept in check at the level where absorption takes place. But then by the time the microbes have moved through the small intestine into the lower intestinal tract, they recover from this viable non-culturable state and they start multiplying again. And that's a good thing, because in the ceca, of course, the number of microbes is extremely high, as Dr. Moore already pointed out to you. So ceca is actually the fermentation vessel of the chicken, where fermentation takes place. And I have presented this already in the previous IHSIG meeting in Sao Paulo, but I think it's a good starting point to refer to when we are talking about the microbes which are present in the ceca, and in the mammals and in humans, and present in cecum and colon. As is often the case of most of the cases, the vast majority of the microbes that are there are firmicutes. Second most important group are Bacteroidetes. And it's more handy, I think, to look at functions of these microbes. And essentially, we can say that most of the firmicutes belong to the short-chain fatty acid producers. They are called secondary fermenters because many times they cannot start fermenting the nutrients that are there by themselves. So in many cases, they need what is called the cell wall degraders, the Bacteroidetes, to help them. Enterobacteriaceae are there as well, and we heard something about it already this morning. And you also have these mucin degraders, and I think mucin degraders are very important because they thrive on the mucus produced by the host. So we should realize that mucus production by the goblet cells in the intestinal tract is not only meant to create a protective mucus layer on top of the epithelium, but it is also meant to feed the microbes. And that is important in episodes when the intestine is empty, in episodes of starvation. Now, talking about the metabolites, which will be important for the host, I think we should think about what comes down the intestinal tract that is not absorbed at the level of the small intestine. That is what becomes available for the microbes to multiply and to produce their metabolites. And first of all, of course, what comes down is those things that cannot be digested by the host because it hasn't got the enzymes available to digest it. And these are the so-called non-starch polysaccharides, which essentially are plant cell wall components. Plant cell wall components which cannot be absorbed because the enzymes are not there, are readily available for the microbes to thrive on. And this is considered to be the physiological situation. So nothing else should come down at the level of the ceca, which can be digested by the host. But unfortunately, in many situations, we see that there is also some of the protein that arrives down the lower end of the intestinal tract. And this is not my finding, definitely not. This is from literature. We see that metabolites produced by the microbes that are present in the cecum and colon of mammals and humans and also that are present in the ceca of chickens, they are based on the metabolic conversion of the non-starch polysaccharides and of the residual protein by the microbes. And so my talk will be focusing on metabolites based on the use of the non-starch polysaccharides and metabolized based on the use of residual protein. Now, this slide I've presented many times before, so for those of you who have heard this talk before, they will be a little bit bored now maybe, but when we look at the non-starch polysaccharides, they have to be broken down to oligosaccharides and monosaccharides, and then they are further converted into different metabolites. And what I want to do now is just to give us an illustration based on work of one of our PhD students who looked at what happens when you provide the oligosaccharides directly to the animal, helping the microbiota to break down the non-starch polysaccharides into oligosaccharides, because this is the hard bit of the work. The hard bit of the work is actually these large polymers which are difficult to access because they are in these plant cell walls which are very dense and very hard to attack by the enzymes produced by the microbes. So if you provide them the oligosaccharides directly, then it may help the microbes to use these. And this is actually the idea behind most of the work done on prebiotics, because most prebiotics are actually oligosaccharides that are non-digestible for the animal. And so the work that Céline did was actually with just adding 0.5% of xylo-oligosaccharides to the feed of broilers, and she could clearly show that this had a beneficial effect on daily weight gain and on feed conversion. So this is not new, because this has been published before. But what we wanted to find out is what exactly does it do to the microbes. And as I liked the discussion earlier on about microbiota in the small intestine and microbiota in the large intestine. Actually, in the chicken, the ileum and colon go together, and they are in the movement and the dynamics of the nutrients passing through the intestinal tract. Ileum and colon are before the ceca. It's contents from the ileum and colon that shift up and down, that are taken up by the ceca and fermented then, and cecal contents is at the end of the fermentation process. Cecal contents is directly passed into the environment. So the colon is actually before the ceca. And that's a bit odd when you think of mammals, where the cecum is obviously before the colon. So colon or ileum, you may consider as one functional unit in the intestinal tract. And in this colon, with these xylo-oligosaccharides, we saw an increase in Lactobacillus ceae, which seems rather logical, of course. But within these Lactobacillus ceae, it was only one species that was increased, and that was Lactobacillus crispatus. And then when we looked at microbiota in the ceca, there was an increase in the cluster 14A strains. This is a family of butyrate producers, and this family was increased, and within this family there was only one species that was increased, and that was the Anaerostipes butyraticus. Now we happened to have both of these strains in culture in the lab, and that's always very handy. So what Céline then did was she took the Anaerostipes butyraticus, so the butyrate-producing microorganism, and cultured it in the presence or not of the xylo-oligosaccharides, and she looked at production of lactate and production of butyrate. And when she did that, she saw that adding the XOS or not did not make any difference. The Anaerostipes butyraticus had a basal level of production of butyrate. So then, looking at the Lactobacillus crispatus, adding the XOS gave a spectacular increase in lactate production, which seems quite logical. But then by co-culturing these together, she could show, she could show that no more increase in lactate because all the lactate was consumed by the Anaerostypes butyraticus and converted into butyrate. And I think this is a very important aspect of the cross-feeding that is going on in the intestinal tract when you look at the breakdown of non-starch polysaccharides. So with XOS, you actually stimulate, you favor this particular pathway. Now, the effects of butyrate I don't have to come back to because I think everybody has been reading the literature about this. There is stimulation of butyrate production is considered to be a beneficial effect. So one of the important messages I think is that one of the effects of butyrate is that it is stimulating tight junctions and by different ways reducing inflammation, not suppressing inflammation, but reducing the need for an inflammatory response more than anything else. Now, the lactate producer, and we were hearing already a number of things about lactate producers this morning. The lactobacillus, in this case, you know, the lactate production increased butyrate production, but it can also increase propionate production by cross-feeding. However, it is clear that when the lactate is not consumed by the butyrate and the propionate producers, there's a problem because quite a, you know, not all, but the vast amount of lactate produced by the lactobacilli and the bifidobacteria is actually D-lactate, and D-lactate cannot be used by the cells of the host, which means that when it's accumulating, it simply results in a drop of pH, and excess lactate will actually make the pH drop from, say, around about 6 to around about 5, and the big challenge is that at pH 5, the firmicutes, which are essentially the butyrate producers, can no longer survive. So you get a dramatic destruction, killing of firmicutes due to the pH drop, and that causes a crash of the whole system. And of course, the consequence of that will be inflammation. Right, going back to our scheme, I didn't say much yet about propionates. Propionate is an interesting metabolite. It's a favorable metabolite, no doubt, because it can be used as an energy source also by the epithelial cells, and it has a number of favorable effects, as indicated here. And one of these favorable effects is that it stimulates satiety, and maybe you're not convinced that satiety should be stimulated in our animals, but I think it should, because when we look at the modern broiler, this broiler has been selected so much towards feed intake that overfeeding is becoming a problem. So at the moment, it looks like producers are struggling more to reduce feed intake by broilers rather than to having to stimulate it. So satiety is actually improving satiety feeling and responses is actually an important objective. Coming back to the propionate production, there has been an elegant study showing the different pathways of propionate production, but what is important to remember is that two of these pathways are only minor in terms of quantity, in terms of importance quantitatively, but this one is quantitatively the most important one, and this one is actually mediated by the Bacteroidetes. And Bacteroidetes, if you ask yourself the question, are these important, are these favorable for the intestinal tract or not? Yes, of course they are very important because they are the first group to attack the fibers. But on the other hand, you should not forget that Bacteroidetes are gram-negative bacteria. And this brings me to a point which I want to stress here, that is that all gram-negative bacteria, as they multiply and die off, and we should realize that dying off of microbes is something that is continuously going on at a huge rate in the intestinal tract. As they die off, lipopolysaccharide is released. And of course, we cannot consider this truly as a metabolite of the microbes, which is the topic of this talk, but I should still warn that lipopolysaccharide is actually a very powerful trigger of inflammation, and it's a powerful trigger also of leakage, of damage to the tight junctions and damage to the epithelial barrier. So excessive amounts of lipopolysaccharide released in the intestinal lumen may be harmful under certain conditions. So we've been looking at some aspects, at some important metabolites derived from the non-starch polysaccharides. But I want to share with you also some thoughts about the metabolites derived from residual protein. And I think residual protein can be important, especially because we tend to overfeed protein to our animals. And, well, I at least have a tendency to eat too much protein as well, because it's palatable. You know, we have special taste receptors for protein, so we like protein, and animals also like protein. So excess protein is something very common. And so when you look at the end metabolites derived from proteins, without entering too much details, this is just a short list of what you can find in the literature. And I want to focus and stress on some of these metabolites together with you. And the first one I want to have a short look at is ammonia. And when you look at the effects of ammonia, ammonia is actually a gas, but in a liquid it will be ammonium hydroxide. And ammonium hydroxide, especially when it's present in relatively high concentrations, it will reduce the receptors for butyrate, which means that production of butyrate will no longer have sufficient effects on the host cells under these conditions. And more importantly even, ammonia will reduce the energy production by the mitochondria, which means that the epithelial cells run out of energy. And the important consequence of this is that the most energy requiring activity of epithelial cells is maintaining the tight junctions between the cells. And these tight junctions constitute the gut barrier. So if the tight junctions are not maintained, the gut barrier is disrupted, and leakage of plasma fluids, but also plasma proteins from the host into the intestinal lumen is taking place, and the practical consequence of that is wet litter. So, so energy production by epithelial cells is very critical, and anything that inhibits the energy production by the mitochondria in the epithelial cells will cause serious damage. So what else is there? Well, there's a lot to do in the human medical world about the biogenic amines, and some of them you definitely know, putrescine, cadaverin, and a number of them. I don't want to go into too much detail about them because they're considered to be very, very important for human health, because they may be involved directly, but probably mostly indirectly, in development of pathology of the intestinal tract, which is typically for humans, and that is colon cancer. So biogenic amines are not of a major interest to us at this moment, but it may be worthwhile to investigate this further, I think also for poultry nutrition. And then we come to the indoles, and the indoles are something very controversial, because when you read the human literature, indoles are considered to be potentially harmful, and we know about a number of these molecules. You've all heard about indole and scatol, and you know the names sound already. When you think of it, it's considered to be not very favorable, because they are also involved in going off of foods and feedstuffs. So they are considered not to be very favorable at least in the conditions where we have been looking at them for mostly. But when you look at the role of indole in the intestinal tract, it actually seems to have a number of very beneficial effects. So indole seems to protect against virulence mechanisms of pathogenic microorganisms. It helps stabilizing those tight junctions, which I was referring to earlier on, and it reduces intestinal inflammation. So indole, although it has a very bad name because it makes food go off, the flavor is actually revolting, but in the intestinal tract, the effects of indole seem to be rather positive. And this will again have an effect on inflammation, actually reducing inflammation again. In humans, as I said, there are effects which are considered negative and even devastating, but these are effects in long term, and we see that especially in humans, the toxic effects in the long term on kidney function and heart function have been focused on a lot in the human medical world. So this is what you get when there's excess indole produced in the intestinal tract. And then Dr. Moore already referred to the neurotransmitters. It's a fascinating area. These are metabolites of the microbes in the intestinal tract, and they are produced by conversion of very specific amino acids into neurotransmitters. The important neurotransmitters that are produced are serotonin, dopamine, and gamma-aminobutyric acid, which are actually neurotransmitters in the nervous system of humans. And that means that these could have an effect on the brain, but they definitely can also have an effect on the neural system of the intestinal tract, which could influence peristaltism, which could have enormous consequences for efficacy of nutrient digestion. So neurotransmitters produced from amino acids may have an important role, but they need to be further investigated. What I want to point out to you also is that the lactobacilli are the most important producers of gamma-aminobutyric acid, and so this is one of the beneficial effects of lactobacilli. Okay, this gamma-aminobutyric acid has a number of documented effects in the intestinal tract, reducing inflammation, stimulating the production of propionate and butyrate, and also under heat stress condition, it has been shown that it has clearly documented beneficial effects. Right, and just to take this one step further, I want to go to another study which Céline did a number of years ago, and that was a study in which we had a relatively acceptable feed formula, nothing special about this feed formula, but what we did is we replaced some of the soy protein by other protein sources which in the industry are known to be less easily digestible. So by only changing part of the protein by a source of protein which is less attacked or less easily attacked by the enzymes of the host, we create a situation where we expect that there's residual protein passing into the ceca. And we compare it to more readily digestible protein sources. And so what we saw is that in the grower phase, the slowly digestible protein, as I shall call it, had a negative effect on daily weight gain and a negative effect on feed conversion. So it means something. So looking at the microbiota, we unfortunately didn't see much. However, when we took this one step further and we looked at the function of the microbiota, we used two qPCRs. One is actually quantifying the genes present coding for an enzyme, which is a key enzyme in the butyrate production, that is the butyryl-CoA acetate-CoA transferase, and the other one is a qPCR, quantifying the presence of genes coding for a key enzyme in hydrogen sulfide production, which is the bisulfate reductase. And as you can see here, the ratio of capacity of butyrate production versus capacity of hydrogen sulfide production was shifted towards more capacity of hydrogen sulfide production. So there was a functional change which actually led to increased capacity of hydrogen sulfide production. Hydrogen sulfide production is a characteristic of the proteobacteria and of the sulfate reducers. So this is what excess protein also does. It stimulates hydrogen sulfide production. And where does this hydrogen sulfide come from? Well, it does not only come from the sulfur-containing amino acids, methionine and cysteine, but it also comes from the sulfated mucins, which are produced by the hosts, and it also can come from sulfides that are present in additives. So we should be careful using sulfited additives in animal feed, because this may potentially increase the production of hydrogen sulfide. But the question is, of course, is the production of hydrogen sulfide, is that a problem? So if we look in our scheme, we should definitely add hydrogen sulfide as one of the effects of protein digestion. But the question is there about the value or the negative effects of this. And what we can find in the literature is that hydrogen sulfide actually has a large number of beneficial effects, which are all listed here, and I don't want to go into detail because that would take too long, but there's definitely a role, a beneficial role for hydrogen sulfide in the intestinal tract. The problem arises, however, when there is excess hydrogen sulfide produced. With excess hydrogen sulfide, you get inhibition of a key mechanism in energy production, which is the mitochondrial cytochrome C. So here again, there is blockage of the energy production in the intestinal epithelial cells with excess hydrogen sulfide. And the consequence of that is, of course, again, problems with the intestinal barrier. So excess hydrogen sulfide inhibits mitochondrial energy production, but it has also a number of other harmful effects. So we should be very, very careful about hydrogen sulfide, because in high concentrations it will again trigger inflammation. There's another little detail which I want to share with you, which I think is, although it's a detail, it may have considerable importance. That is, in these conditions, when sulfate groups are converted into sulfide, hydrogen sulfide is produced, but under these conditions, when there's inflammation in the intestinal tract, this will also be converted in tetrathionate. And for the microbiologists amongst us, this rings a bell, because tetrathionate is actually used in the microbiology lab as a selective chemical to have a selective medium for salmonella. So indeed, tetrathionate allows salmonella to multiply while inhibiting most other bacteria. And that is a problem, because if there's too much protein in the intestinal tract, then you may end up with an animal that will become more susceptible to salmonella. And there's one other organism that is also allowed to thrive when there's tetrathionate around, and that, whoops, sorry, it's missing here, but I can tell you, that is Campylobacter. So an inflammatory condition in the gut with too much protein will stimulate the multiplication of Salmonella and Campylobacter. So one point which I want to address here, but very shortly, is that we should not forget that amino acids can also be converted in propionate and butyrate. And then there's one other study which I like to go through together with you very briefly, and that is a study in which we added a small amount of amorphous cellulose, so cellulose powder, to the feed of day-old chicks, 0.5% in the feed of day-old chicks, and then after that we we switched to 1%. So, you know, with my idea when we were starting this study was this will dilute the nutrients in the feed of the day-old chicks, so you will have a negative effect on performance. But the opposite was true, actually. What we could see is that the birds that got this cellulose in their starter, they had a significantly better weight gain, and that continued during the grower phase. And when we looked at the feed conversion ratio, in the starter, these birds also had a better feed conversion ratio. So what are we looking here? What are we looking at in this case? What happens with the cellulose? It's obviously non-digestible for the host. It enters the ceca. And what happens in the ceca? Well, if we take cecal content and we take it in the lab, bring it in contact with a number of non-starch polysaccharides with resistant starch, and we measure the capacity of the cecal microbiota, you know, as a whole, as a community to produce butyrate, and you can see that some of these non-starch polysaccharides allow the microbiota to produce more butyrate, because this is measuring butyrate production, but cellulose doesn't do anything. So it's not through increased butyrate production that these animals were performing better. So what then happens with the cellulose? It doesn't stimulate this pathway, clearly. And then we looked at the microbiota in these birds, and it turned out that there was just one genus that was influenced. So there was not a major shift in the microbiota, it was just one genus. And it turned out that this genus Alistipes was clearly, significantly stimulated by the presence of the cellulose in the ceca. This is a genus that belongs to a family which didn't sound very familiar to me at the time, these Rechinellaceae. They belong to the Bacteroidetes. And what is known about this Alistipes genus is that it is a strict anaerobe, of course. It's a gram-negative because it belongs to the Bacteroidetes, non-spore forming, but it has a powerful cellulase. So on its own, it can enzymatically break down cellulose. Even if we know that in nature, in the environment, breakdown of cellulose requires a network of microbes, in the intestinal tract, obviously, one single genus can do the job. And then what we also know about Alistipes is that it's present in the core microbiome of the chicken, but also in the core microbiome of humans, and under pathological conditions it can get lost. And also, I like the story, the publication of the group of Torok, and I should be grateful to all the studies which you did, Rob, in Australia with respect to comparing well-performing versus poor-performing birds. Some of these things really popped up in these studies, and now we are beginning to see the light whether, you know, what is actually the meaning of this. So Alistipes is associated with better performance, and its main metabolic end product is actually succinate. And of course, succinate rings a bell to us because that can be used in the Krebs cycle. And we could show in the lab that indeed Alistipes, when you bring it in contact with cellulose, it will grow better, and of course when it grows better, it will produce more succinate. So the cellulose breakdown in the intestinal lumen is the work of a single genus, and what it actually does is it stimulates the production of succinate. But then it becomes a little bit puzzling because it doesn't seem to move further towards butyrate. Although succinate can clearly be converted, at least in propionate, and that has been shown in the literature that succinate can be converted into propionate by a number of different microbes. So this pathway can be stimulated. But what is also remarkable, I think, is that there's recent publications coming out that in the intestinal tract, the epithelial cells have receptors for succinate. So they can take up the succinate from the intestinal lumen and take it up into the epithelial cells. So it can provide the succinate directly to the host. And these receptors are expressed At least it has been shown in the mouse. To my knowledge, it hasn't been shown in the chicken yet. They are expressed in the mouse, they are expressed both in the small and the large intestine on the epithelial cells, and that is just an immunohistochemical image from those studies. And so we know that inside the epithelial cells, the citric acid cycle can use this succinate, but to our surprise, there has been a study coming out very recently with very elegant study using knockout mice, and this study could document that the succinate, which is taken up from the intestinal lumen into the epithelial cells, is not immediately used as energy source for the epithelial cells, but it is converted into glucose, and this glucose is transported into the blood. So this was a mouse study, but I can imagine that this could equally well have been the case in the chicken, explaining probably why we see these better performances. So to wrap up, because I think I've eaten up my time, Mr. Chairman, I think there's a number of metabolites which we know and which we are starting to get a picture of. I think we should also take into account the quantities, because quantitatively there's an enormous difference. Also the power of the effects should be taken into account, and we know of, there are literature data with respect to quantities of a number of these metabolites under physiological and under pathological conditions. Not all of them have been investigated in much detail, but what we should conclude, and that is my last slide, is that some of these metabolites have beneficial effects, clear beneficial effects. Some have clear harmful effects. Some we're not too sure. I mean, lactate is definitely okay, but if you have excessive amounts, you may end up into trouble. Hydrogen sulfide, small amounts seems to be okay, but large amounts seem to be harmful. So I think we should be careful with hydrogen sulfide, and maybe we should put it in the right column. Succinate, we still don't know for sure, but it looks like it may have beneficial effects. And what about methane? I think there's still a lot of work to be done. But what I mean here is, at this moment, we and many other people are investigating the metabolites produced by the microbes. And we know there are powerful tools available which are called metabolomics. And metabolomics are at our fingertips now. I mean, they can produce very fast results as long as you know what you're looking for. So targeted metabolomics are available and they can give fast results. So what scientists need to do at the moment, and what we are doing and many other people, is trying to find out what metabolites should be present in what concentrations to have a favorable effect on the intestinal tract. And my personal view is that I think that within 5 or 6 years, veterinarians will be going to the chicken house. And instead of taking blood vessels and looking at antibody titers or looking at blood biochemistry, they will take fecal samples, send it to the lab, get the next day the metabolomic profile, and by comparing these to the data that have been generated on the normal concentrations of these metabolites, I think they will be able to find out whether the microbiome may have beneficial effects or whether you're in a condition where the microbiome is in trouble. Thank you very much.